We're at the 20th CROI, or Conference on Retroviruses, with John Coffin, who has been past president, or past chair of the conference, and we appreciate your coming back to talk specifically about the basic science elements at the CROI. And it's always a robust area of discussion, and there's probably, I don't know what the percentage of posters, but it must be a very large number of the posters are actually about basic science.
Yeah, it must be 20-25%. I haven't counted them up, maybe even more than that. Touch on basic science issues, including very basic issues, structures of viral proteins and things like that, and going up to sort of basic understanding of virus-host interaction using animal models, for example.
And this year, you maybe can touch on what you thought in the areas we've discussed that jumped out at you as being important, or I mean, nothing here is about clinical science. This is about pre-clinical. So give us a take on what you
I think the potential for using discovery of all of these cellular factors that are called restriction factors, although there's some debate, semantic debate about exactly how you define a restriction factor. I think at least most of us will, we may not agree about how you define it, but most of us know them when we see them. The potential for using those eventually as guidelines or guides to development of novel kinds of therapeutics, I think, is a lot on everybody's mind. Jonathan Stoy touched on that, I thought, very nicely in his plenary talk this morning, for example. We're coming a long way in understanding, and we're always discovering new ones of these, new ways in which our ancestors fought off the ancestors of HIV, and the HIV came back, and the host came back, and the HIV came back, and we're getting a better and better understanding of the arms race that is involved in this, and the complexities of it. We have this we all have this remarkably complex system called the interferon system, where there are hundreds of genes probably involved, and specifically in trying to protect us against HIV and other viruses, and we're just, in a sense, scratching the surface of some of the more obvious features of that. There's a long way to go there.
So it's the host adaptive immune system.
Well, it's the innate immune system. The innate immune system, or sometimes called the inherent immune system, although I think the distinction there is, again, semantic, but that is a major topic of interest now. But the immune system, the adaptive immune system, is still of great interest. We're learning a lot more about the immunology of the virus, and that, of course, has direct application to eventually developing either vaccines, which has proven to be an extraordinarily difficult task for HIV, or some kinds of immunotherapeutic agents, which I personally actually have some interest. So I personally tend to favor as perhaps a long-term solution to HIV infection, maybe.
When we look at the history of the vaccine research, it really does, I think when you look back at the beginnings of the envelope and so forth and how we approached it, it seems like it's so basic. I mean, it's so naive, maybe.
Well, we approached it in a way that would have worked if it had been influenza or polio or any one of a large number of other viruses that we know about, but we came to learn that HIV is really quite special. It's not unique in this. I would say HCV has similar sort of characteristics as far as that goes, and that's another good reason for bringing the two viruses together at this meeting. But HIV was really our first lesson in viruses that have learned over a long evolutionary history of how to make themselves, in a sense, disguised, so the immune system, even in a healthy previously healthy person, cannot figure out how to get rid of them.
Can you address some of the issues about how we can learn from one another at the CROI, cross-pollination, so to speak? And it seems like this is the important part of it.
I mean, HIV and HCV have so many kinds of similarities at all levels, really. Although the fundamental mechanisms by which the virus replicates within a cell are quite different, once you get past that, you have a lot of the same kinds of issues. A lot of the therapeutic development for HCV has really followed the path that was blazed for HIV antivirals. And in a sense, in one sense, they're doing even better because HCV, in fact, does prove to be a curable infection, just using antiviral drugs, which is certainly not going to be the case with HIV. Antiviral drugs alone will not cure HIV. They'll keep it under control for indefinite periods of time, but you can't stop treating a patient and then the virus will come right back again. And that's because of the one major difference between HIV and HCV, and that is that HIV forms a provirus that becomes part of the genetic information of the infected cell, and some small fraction of the infected cells can live essentially forever and probably be replenished. There's information now coming out from a number of groups that the cells that HIV is hiding in over the long period of time some of those cells are capable of self-replenishment, so they can just be maintained at a level. They'll never die out, they'll never decay, because the body just replaces them as a part of the normal way it maintains the immune system for the normal immune system.
We talked earlier about the APOBEC3 and how that blossomed, I guess, many conferences ago, and there's always the kind of a buzz of a new approach, and sometimes we wonder where they are going. And I did see some work on that. I guess it's evolving, but it all takes time. I think
it takes time.
Give us a sense of how that works.
Well, one thing that there was a presentation this morning that showed that one concern we have. Well, let me back up a little bit. The APOBEC3 is a cellular defense, a restriction factor, a cellular defense mechanism that acts by binding, getting taken up in the virus, and then causing mutations in the DNA when that virus tries to replicate. And so the virus DNA that you get, the next step in replication, tends to be what we call hypermutant. It's loaded with mutations, and that just basically kills it. And so in any infected individual, if you look in the DNA in cells, particularly on long-term therapy, you will see a lot of DNA molecules that have had these mutations in them. And the virus has evolved a specific mechanism to deal with that causes APOBEC to be degraded in the cell. It ties up APOBEC and really takes it to the garbage can of the cell. And that prevents the virus most of the time. Some of the times it fails, but most of the time it prevents the virus from being degraded by APOBEC. And it's been thought by some people that this might make a very useful target for an antiviral drug. If you had a compound, a small molecule, some chemical, that would specifically block the action, the interaction between the viral protein and APOBEC, then that would cause the virus to be hypermutated and essentially go away in an infected individual. And it might well work that way. Nobody has a molecule like that, but it certainly would be a fruitful target for drug discovery to try to find such a molecule. But other people have been worried about that because APOBEC mutations might, if it works just a little bit, might increase the frequency of drug resistance, because drug resistance is due to mutations. And there was a talk today that allays that concern to a considerable extent, because it shows when mutations which could have been caused by APOBEC in infected individuals, particularly a mutation to 3TC resistance, M184I, that many of your listeners may know about, that mutation could have been caused by APOBEC, but it wasn't. And when we see that, it's caused in a different way. And therefore, the concern about whether these drugs might have a bad effect at increasing mutation to drug resistance seems to be unfounded. And I would imply that such research might be fruitfully brought forward.
You kind of wonder when you peruse the posters and when you hall talk especially, or at the posters where the presenters giving their background and so forth, that there may be people that are, it must happen like daily that people think, oh, I've got to change the experiment.
Sure.
This will be helpful. I can do it this way. And it's amazing the knowledge that people get. I think this is the reason why we say we continue to have, instead of having everybody at home, it's important for people to be here, that you can get an interaction.
That kind of interaction is critical. That's really what scientific meetings are all about. As you say, we can sit at home, we can read the journals, we can now go, any of your viewers can now go online and see all of the CROI talks, which is a very valuable thing to do, but it's not the same as being here and actually talking to people, interacting with people, and that's where your ideas really come from. This is the source of good ideas. I think is meetings like this.
So, is there anything specific you'd like to bring to the table here that you are working with or working on? That you might be?
Well, one interesting thing we've been working on that interests me, but we don't actually have much to say yet. But I'll put in a plug for it anyway.
Yeah, right.
And we're interested in the interaction between another type of virus called a human endogenous virus, which are viruses that all of us have in the form of DNA that we inherited from our parents, and they inherited them from their parents, and they inherited. And some of these are many millions of years old. And in the humans, there's one group of these that is relatively recently, they were put into our relatively recent ancestors. All by relatively recent, I mean 100,000 years. So it's still a long time ago. And we think there might be some more recent ones around, but we haven't found them yet. If they're around, they're quite rare, but we're still struggling to look for them. But there have been reports in the literature that these might be differentially active in HIV-infected people and might be involved in the pathogenesis of the virus. This is a topic. We have a poster here, which it's really just early days. Well, we show that the expression of this particular endogenous virus, retrovirus, is in fact much higher in PBMCs from HIV-infected people than from uninfected people, and so we think it's a not unreasonable hypothesis that this might actually be in some way involved, in some interesting way involved in HIV pathogenesis and disease.
Is it any kind of a relationship to the way Betty Korber was going back to the beginnings of the first viral that they had? I mean, is that kind of a relatively...
Well, this goes back
I know, it goes
way
back,
before
way
that.
back.
Yeah.
But I mean, is it that same philosophy?
In a sense, yeah. But these viruses allow us. They're really fossils.
Oh, I
some
got
of
you,
them. If we could get some dinosaur DNA, I'm sure we could find them there too. Someday maybe somebody will pull some out of a piece of amber or something. We'll be able to see it. But there are some that we know were fossils of viruses that existed in the very, very earliest mammalian ancestors. And there's some that are found that had to have predated the split between New World and Old World primates, for example, which is on the order of 25 or 30 million years ago. And so they've been around a long time.
It's amazing.
Yeah.
One more question before we leave. The work that you did bringing down to a half a copy of viral load. I mean, you thought, well, let's just do it, I guess, and see what it'll do. But you said at the time it wasn't really important. Maybe it's 50, maybe it's a 70 viral load, and maybe this isn't really important. But I think now it's becoming. Do you not find it's becoming more important?
I think it's important because it's very important as a tool to understand what's going on, what's happening with that half copy or whatever. I don't think there's a biological difference between if somebody is fully suppressed. I don't think there's much of a biological difference in that person if the suppression is to 25 copies or less than one copy. I think that's still about the same thing. It's telling us something interesting, perhaps, about what's happening in that person, but I don't think it yet tells us that there's anything important as far as the outcome.
Right, or a difference between anyone under 70, for instance, would be kind of the same.
Kind of the same.
Yeah.
Biologically the same. Okay. I should point out that I think that assay is going to be more and more important as we're talking about a possibility of a cure, because that will be I think the best way to track whether you're getting rid of the cells you want to get rid of. And the problem with that assay, actually, as Bob Silcano pointed out in his talk yesterday, is that it's very, very close to the background.
Background noise.
Most people, the average number of copies per treated person is about 3. The assay can see down to about 1 the way we do it now. But I think there are ways we can get that assay up to
it's based
by
on
using a much larger volume. This is done with, say, 1 milliliter of blood.
Right.
If we can do that with 100 milliliters of blood, now we're talking about 100, and we can see down. And now if the amount of that virus gets reduced by a large factor, we can see it. Whereas now, in the current situation, if it's at 3 and goes down by more than a factor of 3, we can't see it anymore. So we can't tell where it's gone. And we can't really measure very accurately what's going on. So, so that's actually the first thing on my list when I get back is to start working on getting that assay ramped up, if you like.
Because it's around the corner. They'll be needing that kind of.
And I think that's going to be the assay.
Yeah.
Dr. Silicano's talk really made it very clear that there are very few good ways of measuring what you're doing when you're trying to cure patients and deal with the virus infection in patients that have such low levels of viremia. Perfect
example of what you now have to do based upon what you learned here.
Exactly. Perfect example.
Well, as always, you're really amazing to talk to, and it's always an experience because you only touched on, I think that's the important thing for our audience to know, is that you're just touching on a very small piece of what's happening here at the CROI. And it's all in the literature, it's all in the abstracts, and details that are probably difficult to understand, but you've done a very good job of making it clear, at least to me, for our audience. Thank you very much for doing that.
My pleasure. Thank you.
Thank you, Don.